Yeah. All right. Good afternoon, everyone. We hope you had a wonderful week. It's like getting back in spring quarter week, and welcome to this week's Quantitative Seminar. I am one of your co-hosts, Connor, alongside Angela, and I'd like to first begin with the land development. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Squamish, Tulalip, and Muckleshoot nations. And I'd like to also introduce our speakers for today. John Best is a hydropower scientist at the Washington Department of Fish and Wildlife. He works on mark recapture models for juvenile salmon, IPMs to look at the fish passage survival, and providing quantitative-based advice to managers. He is also a member of the Punt Lab, where he works on spatial temporal models for index standardization. He still owes Andre a few chapters before Sandy O'Neill is a senior research scientist with the Washington Department of Fish and Wildlife. She is the emeritus leader of the Toxics Biological Observation Station, TBIOS. Her research primarily focuses on the relationship between species life history and contaminant exposure and the impact of contaminants on fish health, especially salmon and steelhead. She received her Bachelor's of Science from Memorial University of Newfoundland and her Master's of Science from the University of British Columbia. Please help in giving me a warm welcome for these two.
John and Sandy will take questions at any time during the presentation.
I'm going to just start it off. This is really John's show, but I wanted to thank you for the opportunity to come and present some of the population level modeling that we're looking at for PCB exposure in juvenile Chinook salmon. This project is a collaboration from University of Washington, also Western Washington University, as well as WDFW. Our program, TBIOS, we call it for short, started collecting contaminant information on salmon in 2013. And then in 2023, Andy James from UW-Tacoma and Ruth Sofield, who's up at Western Washington, approached us about doing a collaborative project to look to see if the contaminants that we were measuring in our fish would have some population level impacts. We looked at various sets of chemicals, but what we're going to talk to you about today is just the PCB work. And this project is definitely a work in progress, so we're hoping all the smart minds in the room can provide some feedback. Okay, let's see. I don't have to tell people in this room, what threatens Chinook salmon. There's or salmon in general. There's a whole bunch of things, and in particular, in the early marine survival, there are lots and lots of threats. There was a big effort a few years ago from a consortium of people in the region to look at various threats to salmon in the Salish Sea, and that's what this graphic is from there, where we identified predation and food supply is probably some of the biggest threats to juvenile salmonids, but contaminants was listed as one of the hot threats as well. And that's in part because there are multiple pathways that salmon can affect survival. It can affect them directly, it can reduce their growth, increase their susceptibility to disease, can alter behavior, and also it can be combined with pathogens and climate to have an even greater effect. So based on the Salish Sea Marine Survival Project, we thought, okay, there's some high potential for impacts. There's also a study that was done by Jim Metter at NOAA that looked at coded wire tag returns from rivers that were contaminated versus rivers that weren't. He did this for both Chinook and Coho. And for Chinook, he observed that juveniles that transited through contaminated rivers had a 45% lower return than Coho. He didn't see that for Coho. So whether that's all contaminants go with habitat restoration. Anyway, so that's why we're doing contaminants. It's also my job. So why PCBs? PCBs are toxic man-made chemicals. They're used in things, they're used for lubricants and coolants, so they're in things like transformers. They were banned in the 70s because of their toxicity, but they're quite persistent. They don't break down in the environment, so they're still out there. They're in our sediments everywhere. They also persist in biota. They're not metabolized very quickly. And they also are lipophilic, which means they bind to the fats in your system. And because they're in fat, and when something eats something else, they eat fat, it gets accumulated up the food web. So our ongoing monitoring suggests that the concentrations that we're looking at for PCBs are probably above, levels that are potential health impact. This graphic is just showing you the different isomers or PCBs are like 209 congeners, and they vary in their toxicity. What we're going to be talking about today is total PCBs. Okay, this is a slide just showing you a summary of all the sites that we've sampled throughout Puget Sound. Each of these dots is a river system. So like in the Green Duwamish, we've sampled juvenile Chinook all the way from the hatchery out into Elliott Bay. So these are just the dots to show those river systems. So we've sampled throughout Puget Sound. These box plots here that you're looking at on, this is the right side of the screen, are showing the PCB concentrations in each of these river systems, and we've overlaid this with threshold for PCB effects for both growth and mortality. These concentrations, these threshold concentrations, come from a meta-analysis that was done by Byrne and Tillett, where they generated, based on lab exposure studies, lots of different fish, they generated dose response regressions for mortality, growth, and reproduction. So we're basically using that information, and John will talk about that more. So you can see that the sites that are in red here, they are basically from the Snohomish South. That's where all the development is. That's where all the people are. The little gray here shows the percent impervious surface on the map. So wherever you've got people, you've got PCBs. So we focused for this study, we focused in on Stillaguamish and the Puyallup, in part because we wanted to capture a contaminated site and a non-contaminated site. So did I miss anything? No. So with that, I'm just going to hand it over to John.
Great. Thanks, Sandy. Thanks everyone for being here today. So the goal here is basically to take what we know in between exposure observations. So I've kind of split this into, it's almost a reverse pyramid. We're building up to these population effects at the top here. So we're going to start talking about our exposure observations. And we'll talk about why those need to be converted to sort of a population exposure distribution. And so those are the orange is what we actually have observations for that we've made. The gray here are things we're sort of pulling from the literature. And then the green is things we're synthesizing, the ones below, to get to that. So there's as you can see, there's kind of a lot of moving parts here. So again, feel free to ask questions at any time. Want to try and bring you along with me on this journey. So we'll start by talking about the exposure distributions. So we have they went out and they sampled a bunch of juvenile salmon. I only included the ones from the estuary and the near shore. I didn't consider the upstream ones, just kind of to simplify things a little bit, and because we're mostly we're gonna end up looking at the transition from freshwater to marine. And so that early marine survival is kind of the demographic rate that we're looking to change here.
And almost all the contaminants are accumulated down in the estuary.
Right.
It's very clean.
Yeah.
Upstream for most of our rivers.
And so you can see it looks, maybe we have a nice log normal distribution or something in the Puyallup, and it's just much less in the Stillaguamish. The tricky part here is that these are actually observations of multiple fish at a time. So a lot of times when we measure PCBs, it's expensive. You need a minimum sort of biomass to test. So you'll take the 3, 4 multiple fish, you put them in a blender, and then you're measuring the PCBs in those aggregated fish, or composite sample. And so that's what this looks like. That's what it's hard to see here was the projector. I should have known. But these sort of darker samples are the darkest are individual fish, and then lighter ones. Let's see if I bring my mouse go. There it is. It's somewhere. It's hard to see. This is like 9 fish composited together. And then these dark ones, this is 2 fish and this is a single fish down here. So we do have a few individual fish in the Puyallup, but a lot of these are composite samples. And so if what we care, thank you, that's great.
Thanks.
If what we care about ultimately is what's the exposure of the population, if we're looking at composite samples, those are going to reduce the variability. Those are great if you want an estimate of mean exposure in the population. But if you want to know how many are exposed to very little, how many are exposed to a lot in the population, we need to account for this compositing process. And it's often assumed that the population exposure is log normal. So if we assume the population has a mu and a sigma squared log normal distribution. So if you pick any random fish out of the near shore of the estuary, it would follow this distribution of exposures. I'm using X for exposure here. And then what our compositing does is it basically takes the mean of multiple fish. And so we say, okay, you can approximate the sum of log normals and thus the mean of log normals. Yeah.
So when you say exposure here, is that the concentration?
That's the concentration of
Okay.
PCBs.
That's the, okay, great, thank you.
And so we're in these composite samples, we're observing the mean of these multiple fish. And so if we assume they're all coming from this log normal distribution, we can approximate that with another log normal, depends on how many fish are composited in that sample. And that's, I basically looked it up on Wikipedia and it looked reasonable and seemed to fit pretty well. But you can, there's literature backing it up, of course, but you basically come up with, so if you're compositing N fish, then you get this modified sigma squared and this modified mu. And so plug that into a stand model and you get this distribution of these range of population distributions. And I'll get to what those results look like in a minute. But first we should talk about how we're normalizing these because these are our concentrations. And so the way that they're often originally, the first way that we talk about the concentrations in terms of wet weight. How much does it weigh the whole sample versus how much PCB is in there? A little bit more because these are lipophilic, potentially a little bit more relevant would be a lipid normalized concentration. So how much do all the lipids in the sample weigh versus the PCBs? And then finally, we talk about sort of as our worst case scenario almost is our 1% lipid weight. So if we assume that all of these juveniles are going to get down to about 1% lipids during their outmigration, then that's going to be sort of when they're most vulnerable to the PCBs, because if they're accumulated PCBs, those are bound to the lipids. And as they lose those PCBs, as they're entering the marine environment, they often get down to 1% or so. And that frees up the PCBs to actually be toxic and affect their metabolism and things like that. Yeah.
Just want to clarify, you said they lose their PCBs. Do you mean they lose their lipids?
Sorry, they lose their lipids, which frees up the PCBs.
Okay.
Thank you.
And we don't think they lose the PCBs much at all because they're so slowly metabolized, have like half-lives of 50 years or more. So they do, once they accumulate them, it takes a while to get rid of them.
Yeah.
In 2 weeks or 3 weeks going downstream, it's not going to do it.
So they're getting concentrated as they come down.
Yeah, they gain more and then they lose lipids and so their concentration increases. So we're saying that's sort of our worst case scenarios. If we had observed all of these individual fish at their lowest lipid concentration around 1%, this is the PCB concentration we'd expect to see. And so we fit these models and you can kind of see there's some uncertainty there. And you can see that the wet weight concentration generally is lower. I will say, so I've added the dashed line is sort of our no effect threshold. So most of these, the majority of these fish are not affected by PCBs. And this is a hard threshold and it comes from the literature. And as we'll talk about, there's a lot of assumptions going on here. And a lot of it's coming from the literature, but adjusting any of them could potentially change the results a fair bit. Yeah, Connor?
Are those Bayesian credibility intervals?
Yes, I believe they're 80%. If I remember, it's been a while since I made the plots. But they're curve wise. So it's the collection of log normal densities. So these are log normal densities with each of the fitted mu and sigma values. And then you kind of get a curve wise credible interval here. And you can see, obviously, the 1% lipid fish are exposed, or much more of them are exposed above that threshold. The threshold is obviously different for, that's 0.1 micrograms per gram for wet weight and 22.2 micrograms per gram lipid weight. And then if we look at the Stillaguamish, lower exposure observations, so lower, distributions are shifted to the left essentially. And a lot more uncertainty here because there were 10 or so observations, composite samples in the Stillaguamish versus 30 or something in the Puyallup system. So once we have this population distribution, we want to think about what are the pathways that are affecting demographic rates and things like that. And so we want to look at individual effects. And these, the kind of best source we have for effects of PCBs is a 2019 paper from Berninger and Tillett. It's a meta-analysis of a bunch of different species. And they pulled out effect thresholds and effect sizes. And so the first one we want to consider is direct mortality. So obviously, extra fish dying due to PCBs is going to affect our population. And it's a pretty straightforward log regression. Oops, sorry. And we've got basically following that, it's actually following this dashed line here. The one thing I have not been able to do quite yet was incorporate the uncertainty from this regression into the model. So right now, all the uncertainty in the model is coming from that population exposure distribution that I just showed you. So the next one, so we accounted for direct mortality effects and growth restriction. So smaller fish entering the marine environment are going to tend not to survive as well as larger fish, generally speaking. And so we can get the percent growth impairment from the Berninger and Tillett paper again. They looked at growth, mortality growth and reproduction. We didn't end up worrying about reproduction in this case. But obviously growth impairment doesn't tell us about survival directly. So we looked at a paper from Duffy and Beauchamp from 2011, which found a relationship between size in July and return rates. And we didn't know the size in July of our fish, obviously. So I kind of come at this sideways. And again, these are both like log linear regressions. Or regressions on the log of the exposure, or other way around. Anyways, so growth restriction looks like this. Sorry. Where is my mouse? There it is. Growth restriction is here, G of X. Growth dependent return rate is, I'm calling phi of M. Because that's the size, I'm using M as the size here. And from there we find, so because we don't know what the size in July is, and because the population, the dynamics models that I'm using already have sort of built-in ocean survivals and multiple stages in between. What I did was back out, okay, what would this, given the return rates that we see that are already present in the model, what size kind of corresponds back to that, working backward from the Duffy-Beauchamp relationship. And then from there, we can say, okay, growth-related mortality is going to be the ratio of, let's see, growth-related mortality. So my thetas are all going to be mortality components. And B of M star is the sort of base return rate that the population dynamics model already gives me. Divide that by in this case, I'm doing 1 over G of X. So this is so G of X is how much is growth impaired by PCBs. So 1 over G of X is what would the size be if the PCBs weren't there. So it's the inverse growth because we're removing the PCBs in this case. And then, yeah.
Are they, is this location specific or it's for both populations?
This is going to be specific to each river system, yes, because they each have they have different population models, they have different return rates and things like that. Yeah.
So do you, that figure you were showing for growth restriction, do you have access to those data or you're taking their fitted relation?
I do have access to that. I believe I have, yeah, I have each of these points, yes.
And why not fitting that as a logistic regression rate? Is that not constrained 0 to 1?
That's something that I'm gonna talk about. I haven't tried to fit it as a logistic regression. It's probably a good idea. Right now, we're kind of trying to follow the literature. But I do think that's probably a good next step because when I try to incorporate the uncertainty from these regressions, I end up with things like, more than 100% growth impairment or more than 100%
or negative.
Yeah, right, exactly. Well, so I will say that any of these, that shaded area on the left is below so they show the line there, but the actual rate that I'm using is 0 for anything below 0.1 there. So we got that. So we get growth-related mortality here. And then we want to combine the direct mortality and the growth-related mortality. And we don't want to kill fish twice. So what I assumed was direct mortality happens first and then growth-related mortality. And so the combined mortality is, if 12.6% died direct mortality, then we have 1 minus that left for available for the growth related mortality. And so the red line is what ends up getting used in this case. Now, as I talked about, we kind of have a few different kinds of effects we could look at. Simplest is mortality. We have those, we just calculated them. Survival is ultimately what we need when we're talking about a transition in a population dynamics model. But what we'd really like is what would the population look like without PCBs? We can kind of assume that the population models that I'm using already incorporate some of these PCB effects in them because they were built on these populations that are already affected by these PCBs and have been for decades. And so what I actually end up looking at is 1 over the survival, because again, that's just like I mentioned with the growth change. That's sort of what would our survival look like if the PCBs weren't there. And so this is going to be a factor change in our demographic rate. Now, we have separate population dynamics models for each system. We actually have 3 because we have 1 for the Puyallup and 1 for the white, and those are separate. And then we have another one for the Stillaguamish, I'll get to in a minute. So the Puyallup and the white were both derived from the EDT model, the Ecosystem Diagnosis and Treatment model, that was developed for it a number of years ago. It uses almost all density dependent survival functions, but a lot of them, like the marine survival and some of the others, have such large capacities that they're essentially density independent. We dropped the yearling migrant portion of the white river model because it's something like 1% of returners are yearling migrants. And it was a lot of complication for very little, what was going to end up being very little difference. And in particular, what we're looking at is this transition from essentially from freshwater to the marine environment. And that's where we're adding this modification to the demographic rate that we were calculating. Similar story in the Stillaguamish. This one's derived from the HARP model developed over at NOAA by Tim Beachy and folks. And again, it's fairly, these are all very much simplified versions. These are both originally intended to be sort of spatially explicit habitat models. So you could say, oh, if I do some restoration work in this reach of the river, you know, how much does that change the population? And so we kind of smushed everything together, integrated over the different reaches and things with help from the folks who developed the models to get these population dynamics models out. All of these models do have density dependent stages, which means we can end up finding stable populations in the end, which is going to come up later. And again, we're looking to change the demographic rate as these fish go from the freshwater to the marine environment. Now, to get those demographic rate changes, we need to combine our population exposure, how many fish, you know, what proportion of the population is exposed to 100 or one microgram per gram PCBs versus 2 and things like that, and combine those with our individual effects. And so again, we're looking at what populations look like without PCBs. So we're saying what's 1 over the survival, and that's going to be our increase in the survival from freshwater to the marine environment. And to get that, this is our increase in survival of 1 over 1 minus theta, and then multiply by our population exposure distribution and integrate that, you get an expected survival for that. And then do that for every sample that I got, every value of mu and sigma that I got for those population distributions. And I get a distribution of these factors. So it's sort of trying to at least propagate some of this uncertainty through. And that gives us this. So these are, ultimately, we're only looking at the red effects, the combined effects, when we look at the whole population, but it is interesting to kind of see how much is coming from direct mortality versus growth-related mortality.
So it says is 100% here, no change? No
Exactly.
effect, but
Yes.
it's not 100%.
Yeah, this is sort of the factor we're multiplying the.
All right.
Yeah, we're not.
It's relief.
It's still quite high, it turns out, probably higher than we would expect it to be, but especially once we see the population results. But yes, so this is the 1.05 times the existing transition rate in the dynamics model. Mark?
So sorry, this might actually be a question more for you, Sandy. Is the idea being that burden fish have lower growth rates and therefore more susceptible to predation? Is that where what's the PCB effect on mortality?
Yes, the PCB effect would reduce the size, and then smaller fish are more likely to be eaten. Is basically what it comes down to, right? Is that what you're asking? Say that?
Yes.
Yeah.
That would be the hypothesis.
And you have data on that because there's coded wire tags, so you can tell
Well,
small fish come back as often.
there's variable data on it. Like when this Duffy and Beauchamp is a really nice relationship, and like, is it real? You know, space she pooled together salmon in Puget Sound that were caught, and she looked at the coded wire tags for those populations and went back and, you know, did an analysis. It's the Buffy and Duffy and Beauchamp is showing, like, can you just go back to that? It's a really high level of increase. Sorry, John.
There it is.
Yeah, so you're going from, if you go from 10 grams all the way up to,
It's 10 grams to
yeah,
40 grams.
yeah, you're basically going, you're tripling the survival. It's going from 0.4 like to 2. So that's pretty high. And there are lots of Zabel has done, work on showing good relationships. Across multiple populations, within a population, not so much, so. There's a big chunk of uncertainty there. We were using this one because it was local and was available to us, but. You'll see later, that's a caveat. We
It
don't-
look very good to me.
Huh?
The data don't look very accurate to me.
Yeah.
I know, but it does,
Pretty darn good.
it does, but it's, but it's one study, so, right?
Yeah.
Sorry, John, did you go through all that?
That's great. Let's see, we were here, so we just did the integral and we got to these sort of demographic rate changes, so. Up in our worst case scenario, we would see. In the Puyallup or the white, we would see almost, the survival would be 20 from freshwater to marine would be 20% higher. If PCBs weren't present.
And we should point out that the Berninger and Tillett paper was based on wet weights of
Yes.
PCB concentrations. And so when we tried to come up with the threshold for a lipid weight, we used an average lipid weight for the fish in the experiment. So it was a very crude correction. So making the leap in those regression curves from wet weight to lipid weight has a bit of uncertainty in it as well. So.
Yeah, thanks. Okay, so now we have those demographic effects and we have our population dynamics model. We can put those together and see how it actually changes the population. To get to that, I ran a population model, and because there is some density dependence in there, you can just kind of iterate them until you get to a stable state. And we decided to use number of returning spawners as sort of our primary metric to look at. And again, this incorporates the uncertainty that was propagated from the population distribution parameters, and which gave us uncertainty in that change in nearshore survival. And now is giving us uncertainty in the population of that. And that ends up looking something like this. So this is the combined Puyallup and white return spawners. And we got, with just wet weight, we get about a 4% increase in spawner abundance if PCBs weren't there. If for lipids, we got about 38% and 1% lipids, 51%. Now these feel far higher than they are. These don't feel particularly realistic to us. And like we mentioned, there's a lot of assumptions, there's a lot of approximations in here. And so we're still kind of working on those. Stillaguamish with much lower exposure under wet weight saw essentially no effect. I think the top of the 95% credible interval was like 0.5% change. So I can't imagine we'd be able to detect that in the real world with normal variation. 1.6% increase with lipids and 5% with 1% lipids.
So yeah, just trying to wrap my head around why there's such a big difference between these three. Like these numbers are based on studies which are based on lipid concentration level. I mean PCB concentration levels. The measurements here are just different ways of measuring the lipid, the PCB level.
Yep.
So in some sense,
Right, but
I guess what I'm struggling with is you're just measuring the amount of PCBs in different ways. So why does that influence the survival?
well, I think the relationship when we convert to lipid weight, the relationship in Berninger and Tillett is all wet weight. And so he used an equation in his model where he had some lipid weight data and he converted it to wet weight, and in order for him to do that, he said, oh, we'll use the average lipid weight in our studies. And of course, fish in exposure studies are fed on a full ration and they always have higher levels of of lipids.
Uh,
So the average in his studies, I think, was 4.7%.
That sounds right.
So I don't remember exactly how we, yeah, so how did we?
Whereas reality fish have much less lipid, and so the.
Oh, yeah,
actually-
yeah, so
Okay.
even hatchery fish, when they come out of hatchery, might have 4 or 5% lipids, but by the time they get downstream, they're in the low twos, and even hatchery fish will get to 1%. So what we see in juvenile Chinook is really, really low lipids, and so we think PCBs can have a pretty strong effect. So, if PCBs are lipophilic, they're locked up in all the fat, like whales, right? So it's all in their, and it's in their blubber. But if they're starving and they're losing that fat, there's less places for that lipid PCBs to sequester, and it circulates more. So in juvenile Chinook, they're like teenagers, they're growing like steak, and they have no lipids, and so the PCBs are circulating around. But whether or not the burnage or intelligent transition that we're making in that may be a problem.
Okay.
So that's, I think that's one of our uncertainties. Would you believe the wet weight? That's our safest, because that's what's published, and there's less conversion. So if you went back to the Puyallup, like 4%, PCBs, PCB is definitely one of the more toxic chemicals we look at, so I don't know. And if Jim Metter was seeing up to 25% reductions in returning adults in contaminated rivers, so that doesn't give me any pause, but when I see the 38% and the 51, I think, is it our assumption, or is it something to do with the population models? And then I don't know.
Yeah, to be determined. One thing that we ran into here that I thought was interesting and a little counterintuitive, and I spoke with one of the folks who works on the heart model about it, was starting from these effect sizes where we're at, even at a 120% increase in survival in that single stage transition, we end up with a 50% increase in the number of spawners. And I thought this would be a good group. We kind of intuitively would have expected that, because of density dependence, that effect would have been attenuated rather than amplified. But we confirmed that sort of the one variation of the heart model does sort of do that as well, where it kind of, you get almost a doubling of the effect. At least for this, I didn't try changing a bunch of the different transitions, but.
Is that because of where it's occurring in the life cycle?
Possibly, if it's that near shore, the early marine transition, and then they're mostly density independent until they get back to heading upstream, and then I think usually, upstream is density independent, and then eggs, like laying eggs, and that becomes density dependent again.
I just had one question about the seasonality of exposure to PCBs. If that's, if there is a strong seasonality to it.
These samples are all collected in the spring, when the fish are going down river. So these fish are all losing lipids. So we're not getting them at other seasons, right? If you look at PCBs, say in resident Chinook salmon, if they leave and they stay in the Puget Sound, we've sampled those over time. And what you'll see there is the lipids are quite high through the fall and early winter, and then starting in like late February, March, they really drop off, and so you get low lipids. I think we have a little bit of data from juvenile Chinook salmon that were caught in the central Puget Sound in February, March, and in those cases, like as these fish leave the river system, they come out of the Green Duwamish or the Puyallup, they're pretty contaminated. And then the longer time they spend in the central Puget Sound basin, they will actually continue to increase, like continue to pick up PCBs, but the concentration will go down because they're feeding on more cleaner food. Whereas in places like the Skagit, they're coming out of there And they actually are getting dirtier because the scadge is pretty clean, but there's some PCBs in the central basin, and so their concentrations go up, right? And so what we notice though is the lipid levels in the winter months, so if it could even be worse because the lipid levels go down to about 0.5%, not even 1%, 0.5%, and they, the PCBs are still going up. So potentially, a stronger winter effect, but so how we're modeling that lipid in this model, I just don't know.
Yeah, and in this model, it's all in that single transition,
Mhm.
life stage transition to the marine environment. So there's not really room in the model for seasonal. If we talked about potentially, say here, including a reproductive effect. So that would be in our sort of spawner to egg transition, would reduce the number of eggs from individuals who are exposed to higher concentrations. But the Berninger and Tillett paper didn't have any salmonids in their reproductive effect meta-analysis. And it was many fewer. It was based on fewer than 10 papers, if I remember correctly. So the data were much sparser. It was going to end up adding up, more dynamics, and then we want to think about, like Sandy said, fish who are resident to the sound continue to pick up PCBs. Fish who head out to the open ocean are not going to come back with as much of burden.
Or even the juveniles that are gonna leave the sound after that first winter, right? We just got them as they come into the sound.
Yeah.
So by February, a lot of them are gonna be out of the sound. There'll be some that will reside, but so the model, if it was working well, we could do other life stages, but we're still on stage 1. So,
I think this is not a criticism of York because all this other stuff did it before, but all that downstream later time mortality is all additive in these models, right? There's no compensatory. So,
Yeah.
right. I mean, that's the way it is, and that's gotten a lot more attention. The question is, would most of these fish have died anyway?
Right.
Maybe PCBs is driving them lower, but yeah, then they gotta swim out through that gauntlet of harbor seals and everything else.
Yeah.
Toast, so.
Yeah.
Somewhere down the road, I think it would be interesting to use some of the hit tag data to see if it's saying anything about where the sources of mortality were.
Yeah, absolutely. So we looked at these. So we talked about a few. I'm kind of coming to the end here, but we do have a bunch. Obviously, this is sort of assumptions all the way down almost. There's a lot we're working on based on other people's work, and conversion factors and uncertainty and things like that. So just to be clear, the Berninger and Tillett meta-analysis is kind of the best we have for PCB effects. It is a meta-analysis based on, and includes a few salmonids in there. The way that they measured effect sizes maybe isn't the most appropriate for what we're using it for here. I think it was something like, I can't remember.
what's the difference between the control versus the treatment?
Yeah,
Was it, and they use the relative effect, right?
Right.
It's 10 times higher in the control. So that was the number that went in the model.
yeah. So there's some concerns about that. Again, some of these without, a whole separate research effort, we can't do a lot better than what we have right now. We mentioned the uncertainty converting wet weights to lipid weights and lipid weights to wet weights, and how that's it's a little crude right now. The Duffy and Beauchamp size survival relationship. We mentioned there's probably additional uncertainty in there. There's probably and trying to, I had to really kind of go roundabout to get something I could use out of that relationship, because we don't have July size in July and we don't use return rates directly. So it was a little roundabout, and obviously that introduces lots more room for uncertainty and things like that. And then life cycle model parameters, these are based on these habitat based models that maybe aren't ideal for this kind of work. It would be interesting to compare this to, say, a more traditional IPM. And kind of try to plug these effect sizes into a more traditional IPM. That includes uncertainty and things like that.
There was another effort by Jessica Lundin at NOAA to apply a life cycle model down in Portland Harbor to Willamette juvenile Chinook. And if I'm remembering correctly, they saw it's like 54%, was it, reduction in survival? But that only translated, associated with PCBs, and that only translated to a 20% increase in the number of returns. So we haven't looked at that model in detail versus these life cycle models, like why are they giving us such different numbers? I do believe that Maya, correct me if I'm wrong, Maya she did sort of test each model in this study against each other. So she took the PCB levels in the that we observed in the Puyallup and applied them to the HARP model in the Stillaguamish, and vice versa, where she took the low concentrations in the Stillaguamish and applied it to the EDT model. And they both basically gave us similar results. So that gave us some sort of a comfort that maybe these models were telling us the same thing, but
It's the PCBs driving these changes rather than differences in
the model.
the population, yes.
Yeah, but we haven't looked at this other life cycle model. I believe it was probably the guy's name. Anyway, someone I know another life cycle model. So
Great. So to kind of keep pushing this forward, I'd like to incorporate more uncertainty. I think going with something like a logistic regression rather than sticking with their linear regression, it's not going to extrapolate. You know, their linear regression works fairly well because they're not trying to extrapolate out into the tails of the population exposure like I'm doing here. And what that ends up with is I get estimates of zero survival, which of course gives me the factor of change of that of that nearshore survival becomes infinite. So that doesn't work for obvious reasons. And so moving to logistic regression and maybe trying to kind of clamp down things a little more realistically that way, I think would be a good way to move forward. That's a super helpful suggestion. Thanks, Mark. And then considering sort of why are these life cycle models behaving the way they do? Why is it amplifying these effects rather than attenuating them? I think that's worth kind of trying to figure out, and it's not what I expected to see. And I'm not quite sure why that's happening. So I'm curious about it. So we were able in some fashion to take these contaminant effects and scale them up to a population level change. Obviously, we've got to be careful and explicit with our assumptions because this is all built on those assumptions. There's a lot of uncertainty to account for. We're hoping that, you know, with some further work, this will be able to be used for monitoring populations and prioritizing habitat projects. You know, is remediating PCBs going to be more valuable than, you know, putting some more large body debris upstream? I don't know at this point, but you know, hopefully these are the kind of questions we could start to get at. Sandy mentioned this is a big collaboration, but it was we also got help from a lot of outside folks. Greg Blair at ICF was one of the primary developers of the EDT model that we pulled the Puyallup White from. Ariana Timbici, Corey Green, the team at NOAA did either toxics work or developed the HARP model that we use for the Stillaguamish. Puyallup tribe did a lot of the sampling. I believe that was probably what the Stillaguamish tribe also did. Stormwater Center is at UW Tacoma, and a few other folks who helped us at DFW. Does anybody have any questions?
Should we give up? Keep going.
So anyone online can raise your hand if you'd like to ask a question. I can unmute you, or you can just put it in the chat. Are there any other questions in the room to start though?
Yeah. I mean, an experimental question, all the salmon that are getting sent out have, a lot of them have coded wire tags. Is it feasible to take a sample of PCBs and then see how many of them survive based on the amount of PCBs in the outgoing salmon? That would give you a direct measure of survival.
That would be an awful lot of sampling that you'd need to do for,
It'd be expensive to do the PCB.
oh yeah,
Yeah.
they used to be $1,000 a sample and now it's $1,500 a sample. So that's why we are doing, there are some really good exposure studies that have also been done recently. You expose them in a pen kind of thing and look for reductions in growth. So there have been studies done at NOAA going back into the mid-80s where you can like take fish from the Nisqually and expose them to waters from, the Green Duwamish, and you can see a 10% reduction in their growth. So, there's definitely you can see laboratory effects for these things, but getting them at the population level is really, really high, and even like if Cote d'Ivoire tag group, we had some fish that were like 50,000 fish that were tagged in a Cote d'Ivoire tag group, and they were like delayed release fish. There weren't enough caught in the fishery to do any expansion, so it's hard to do.
There's also a minimum, like you need an amount of biomass to actually
Yeah,
run the
so
state.
we need a minimum of 2 grams of tissue for PCBs, which doesn't sound like a lot, but if you've got a little, natural fish wild fish coming down, like you're lucky after you remove the otolith and take out the gut contents and the food scale, you're down to like a gram, right?
You can't
So
sample them in the
Yeah, so that's
the front half and the back half of the fish release.
Yeah.
That's the survival.
So, but, I do think we're, like I said, I think there's a lot of work showing there's an effect. We're just trying to get a handle on what level it is at the population. I think the only way to do it is to monitor this. We can't, that's what Jim Metter was getting at, though. Can you look at returns of Cote d'Ivoire tag returns of fish going back to the Puyallup versus those going back to the Stillaguamish? And when Jim Metter did that study, I'm sure he did not see an individual river effect, which is why he pooled all contaminated rivers and non-contaminated rivers. And that may be a factor with sort of variation in marine survival as well, right? Over time. So you can see it in developed and undeveloped systems. I think Arnie Magnuson and Ray Hilburn also had a study with Chinook and Coho with some coastal populations where they looked at Cote d'Ivoire tag returns in developed and non-developed estuaries. So not necessarily contaminants, but if it's developed, you can have some contaminants. And they were seeing similar types of reductions in survival in these developed estimates that Jim Metter was seeing. So the combination of contaminants and physical degradation that happens in these urban centers is real, and it's probably up maximum somewhere around 30–40%. So that's why we don't believe these 51 and 31% for one contaminant. It is one of the more toxic contaminants we're dealing with that we know about here for like Chinook, but still they just don't seem real. So there's something in there that we're not doing right.
Yeah.
I'm asking you to go back quite a few slides to your earlier bits to the contaminating.
Population exposures or the effects?
Keep going back.
These.
Yeah,
Yeah.
so those curves are coming off of fits to, if you go back even further, the observations, right?
Fits to these, yeah.
Yeah, so do you play around with these, like pulling some observations out, like that one that's way out there on the right? And how influential it is?
There's actually 2 out there.
Okay,
But yeah, some leave one out validation would be would be a good idea. I think this is honestly sort of a separate paper that I'm interested in developing. Because there's more interest in sort of population distributions of exposures in toxicology, I think. And a lot of the work is done in composite samples because you need a minimum size and they're expensive. So if we can disaggregate these samples, that's gonna be useful for a lot of folks.
There was one study with Jim Metter and others at NOAA where they looked at individual fish. They had a lot of individual fish. They collected them a little later in the summer, so they were bigger and they could get more tissue. And they had, again, a lot of normal distribution, lots of fish with hardly anything in them, but they had everything from nothing up to, thousands of PCBs. So you do get these hot individuals for whatever reason. They're picking up some contaminated gunk on the bottom. They're hanging, I don't know. So we even occasionally get some contaminated fish at hatcheries. Like we collect the fish at a hatchery and they're clean, and every now and again we'll get one that's like, instead of being 0 to 2 or 3, it's 250 nanograms per gram. And so we think it's possibly, is it like the paint in the raceways have PCBs in it and it's chipped off and the fish is eaten a bit, and we have no idea. We have no idea. So it happens. But there is a huge amount of variability. We do have one exposure experiment that we've just finished with Ruth. It was a short-term exposure study to look at stormwater, but we took fish that had been held offshore at Point Defiance, in the outer parts of Commencement Bay, and we took them from there. They left some at Point Defiance, and then placed others of them in other areas, like transplanted them into little net pens in Puyallup, which is our most contaminated site, and a couple of others. So we've sampled those now, we've got them all processed. So we're looking at those for growth rates. And then we're gonna, depending on the range of growth rates we see, we're gonna pull the individual fish that were held and measure them for various contaminants, won't be just PCBs. So, but none of them are cheap, right? Yeah, so paying John to work on this was nothing compared to the chemistry.
So what is the long-term trend in PCBs?
Oh,
Over decades. I mean,
good question.
Get around the people that was banned in the 70s?
Yes, banned in the 70s.
Is it not just ending in the sediment and disappearing from the system?
So no, well, it gets resuspended in the sediments, but it also gets in the food web. And so if you think of it, it gets into the food web. If the plankton die, they fall, they become sediment. But if the plankton get eaten by, euphausid that gets eaten by a heron, basically it gets recirculated in the food web, right? So that's why they hang out for so long. But globally, there was sort of a drop off in PCBs post, through the 80s, and in many places they've leveled off. So we've gotten data now that NOAA used to do a lot of monitoring, and we took over in 2013. So we've gotten their historical data and our data. Methods have changed over time, so we've been working on that. And so it looks like in many places there have been a decline in PCBs at some of the worst sites, like in downtown Seattle, Duwamish. There's a place called Slip Floor. It's really hotspot. They've done a whole bunch of dredging and cleanup, and that's getting better. But there's another place across the river, Kellogg Island, right in Harbor Island, and that's less certain that has gone down. That there's no obvious trend there. So we're working on those. Hope to get that paper done soon. So there's some declines in things like herring in the Sound. We've seen declines in PCBs. But they've, not great declines in PCBs, they've leveled off. So PBDEs, which are newer, those have dropped off more. So they're still out there.
Yeah.
I believe Paul Chitter at NOAA was looking for declines in southern resident killer whales, and I think he said that based on the biopsy samples, they were starting to see a decline. I think, but I haven't seen that data published, but.
Okay.
So it's slow to come in.
Just keep having babies going floating them.
Yeah.
Yes, I was gonna say. We'll say, so the work that the T-Bios group that Sandy headed up for a while. Does is super interesting because they use there's estuary. Near shore, so it's juvenile salmon, mussels.
Yeah, so we
And
sample the
here-
whole of the ecosystem, so we have. Our indicator species for the estuary, river estuary is juvenile Chinook, and then we place caged mussels in bay mussels in the near shore, so in the winter, and then we have English sole as our benthic species, and then we have herring for the pelagic work, and we also have adult salmon in the pelagic food web, and we're definitely a PCB hotspot on the West Coast because our, Puget Sound doesn't exchange very well with the ocean. So a lot of the contaminants that get in the sound stay here in part because a lot of our species are also resident. They don't leave. So, yeah, so it's a bigger program, but we just showed you some of the juvenile. So we are seeing trends, declines, in other species, but they're slow. They're not rapid.
Yeah, great, thanks everyone.
Wonderful, thank you.